Hello, Instandard. Have a good day. Have a nice day. I hope you are doing well. Today we are going to do video number 7, lesson chapter 9, reproduction in animals as part 2. Okay, video number 7, chapter 9, reproduction in animals part 2. Okay. As I say, this part 1, part 2, the portion of the lesson I am splitting. So just for our reference, when you are going on to the video, we should know the content is enough. So with that we will understand part 1 we have learned this portion, asexual reproduction. Part 2 we are starting with sexual reproduction, and next we are completing that sexual reproduction. Like that, for our understanding I am giving it as part 1, part 2, part 3. Otherwise, it has nothing to do with the lesson. I hope you understand. So, today, video number 7, chapter 9, reproduction in animals. Right? In previous session, we have learned what is reproduction. That is perpetuation of the species. Spelling is check, perpetuation, or continuation of the species from one generation to the next. Are you okay? Are you clear? So with this reproduction, we have learned as asexual reproduction and sexual reproduction. What's the difference? Asexual reproduction, only single parent is involved and no formation of gamete and no variation will occur. Whereas sexual reproduction, there is involvement of both the parents, male and female. As a result, new gametes are formed again as male and female, and then as a result of this, further variation will occur. So that is sexual reproduction. And then in asexual reproduction, we have learned about what? As fission, as binary fission and multiple fission, and then budding, example hydra, fission example amoeba and paramecium, and plasmodium, malarial parasite. And next one we have learned about regeneration in planaria, and lastly as budding in hydra, by budding in hydra, that we have learned. I hope you understand. And then we learned about metamorphosis, where there is a series of changes from egg stage to the adult, and it is totally different. Egg to adult, there is transformation. That phenomenon is called metamorphosis. And there are two types: incomplete metamorphosis, example grasshopper, and complete metamorphosis, example frog and butterfly, that we have studied. I hope you remember all that. And the technical terms we have learned in that hermaphrodite, where the presence of both male and female sex organ as testes and ovary present in the same animal. Example again hydra. In that condition, when testis matures first, it is said to be protandry, and when ovary matures first, it is said to be protogyne. How are you to remember this? Andry referring to androecium, male part of the flower, gyne referring to gynoecium, female part of the flower. As it matures earlier, we use the term pro. So, protandry, protogyne. I hope you understand. You can recollect all this. So, this is for asexual reproduction and metamorphosis. Today, in video 7, we are going to learn about sexual reproduction. We are going to learn sexual reproduction. As before we enter into that sexual reproduction, we come across the term chromosome. We should learn the term about chromosome. Right here, chromosome, and then mitosis, meiosis, chromosome, mitosis, meiosis, diploid, haploid, haploid, and then we say about the DNA and genes. Okay, and then this is the sex organ, testis, ovary. I hope you follow this. So here we come across the term with reference to sexual reproduction as mitosis, meiosis. These two are cell division, right? And then diploid and haploid, that refers to chromosome number, that we will learn now. Okay, and then DNA, deoxyribonucleic acid, and genes are the carriers of hereditary characters, and testis is the male sex organ and ovary is the female sex organ. I hope you understand. So we say during sexual reproduction, male and female sex organ, male and female, these are the parent, male and female parent. Okay, and male will produce sperm and female will produce ovum. Ovum or egg. These are called gamete. These are called gamete. Okay, and as a fusion, this is fertilization. This is fertilization. Fusion of male and female gamete is called fertilization. And as a result, zygote will be formed. As a result, zygote will be formed. I hope you understand this. Now I say male parent will undergo the process. As I said, when the child is determined, the sex of the child, when it is determined as male, testis is the male sex organ, and that will grow, develop till the age of 12, and then from the age of 12 to 15, it starts maturing, and that will undergo the process for reproduction. Same in female, when the sex is determined as female, the development of the ovary sex organ will start develop, will continue, and then at the age of 11 to 15, it will mature, and that maturation will release the ovum. Age attainment female. Otherwise, we say they attain puberty. They attain puberty. That is maturation. I hope you follow that. Now, during that time, we say in human there are 23 pairs of chromosomes. There are 23 pairs of chromosomes. Okay. Now, these 23 pairs, they occur as 22 plus 1. As this 22 chromosomes, they are called autosome. Autosome. And this one is the sex chromosome or allosome. I hope you understand. It's not given in our textbook, so please understand. Listen carefully. The chromosome in human, there are 23 pairs. Now these 23 pairs, they are occurring as 22 plus 1. What way? 22 autosomes and 1 anosome. Otherwise, we say 22 body chromosomes or somatic cell or vegetative cell, and one is present in the sex organ. That is called allosome. I hope you follow this. Allosome. Now we say, next lesson we are going to learn plant cell and animal cell structure. Now when we take any part of the body, when a small scratch is happening, and that can just take a small piece. When you see under the microscope, we find cell structure. Okay, and that cell structure, every cell will contain 23 pairs of chromosome in human. Okay, and all these 23 pairs, they will take part in cell division. Now we say the child grows, is it not? And that is by the developed multiplication of these cells. And these cells will undergo the process of cell division, that is called mitosis. I hope you are listening. So now this mitotic cell division will take part in all the cells, and when at the time of reproduction, the sex chromosome, the sex cell, testis and ovary will take part in the process of reproduction, and that is for the formation of sperm and ovum. And during that time of cell division, meiosis will occur. So that's why we say mitosis, it is indirect cell division, and meiosis, it is reduction division. And another chapter. Okay. So now when any cell, for example, here male, it is testis, and here it is ovary, and that will undergo the process mitosis, and then it is meiosis. And that meiosis cell division, it is called the gametogenesis. It is known as gametogenesis, as spermatogenesis, and oogenesis. Spermatogenesis taking place in the testis, and oogenesis will occur in ovary. Okay, and during that division, it is the formation of gametes, and the meiosis will occur. The cell division called meiosis will occur. I hope you understand this. Now during that time, the chromosome number, we say 23 pairs. Yeah, we call it as 2n or diploid. We call it as 2n or diploid. Why 23 pairs? In that, one from paternal mother and another from maternal mother. This is for the mother. So one from paternal, one from maternal. So every body cell or somatic cell or vegetative cell will have diploid number of chromosomes. That is represented as 2n. I hope you understand. And that diploid chromosome, during the formation of gamete, it forms the haploid gamete. N. This sperm and ovum, it is haploid number. So we say from diploid, it is reduced to haploid. That is called reduction division. The chromosome number from diploid to haploid, when it is reduced, it is called reduction division. So, as a result, this is diploid to haploid. So here testis on the diploid. Testis and ovary, it is diploid and that is reduced to haploid. So it is called reduction division. I hope you understand this. So now we say the chromosome number from 23 pairs diploid, it is reduced to haploid number as sperm and ovary. Ovum. This ovum, it is also known as egg. Then at the time of fertilization, fusion occurs and as a result zygote is formed. So we say sperm n number haploid, ovum is also n number. Now this diploid zygote is formed. Like that the cycle repeats. I hope you follow this. So this is for the chromosome. Before we go for the sexual reproduction, we have to understand. What are all? First we say sexual reproduction. It is first reproduction, perpetuation of the species, asexual and sexual. In sexual reproduction, the sexes are separated. That is called sexual dimorphism. When the sexes are separate, it is showing sexual dimorphism as male and female separate. Okay, and they are having diploid number of chromosome, that is 2n. Diploid number, one from father paternal, another one from mother maternal. So when this parent, male and female, with the male sex organ testis and female ovary will undergo the process gametogenesis. As sperm testis will undergo the process spermatogenesis and ovum will undergo oogenesis. Okay, so at the time, first the division of this testis and ovary cell, it is mitosis. Then at the time of gamete formation, it is meiosis. I hope you follow this. Thereby, diploid number is reduced to haploid. Okay, and then fusion occurs. That process is fertilization. I hope you follow. So that is for reproduction. Then we will continue this as in our textbook. What is given? 1 minute. Testis. As yesterday we have studied about sexual reproduction. Next we will learn about structure of male and female organ. I hope you follow this. And then we are talking about external and internal fertilization. So this in human it is internal fertilization and viviparous. What do you understand viviparous? Giving birth to young one. Okay. So viviparous, giving birth to young one. Other one, oviparous, we have learned. Is it not? So viviparous, giving birth to young one. All the mammals, they give birth to young ones, except platypus and spiny anteaters, they are egg-laying mammals. In that, another one, oviparous, egg-laying animals are called oviparous animals. In that, we come across a term cledoid egg. We come across a term cledoid and non-cledoid egg. Cledoid and non-cledoid egg. The eggs which are laid on land, they are covered by a hard calcareous shell. They are called cridoid egg. So the eggs which are covered by hard calcareous shell, they are called cridoid eggs. Whereas the eggs which are not having calcareous shell, they are called non-cridoid eggs. Normally, these non-cridoid eggs, they are laid in water. Example, fish, frog, and other aquatic animals. Okay. So when this fish and frog, they normally lays hundreds of eggs, and those hundreds of eggs, they are clumped together. They are not occurring in single. They clump together. That is called the spawn. They clump together, called spawn, with the jelly-like mass, because they should not be spread away or carried away by water easily if they occur in single. And then as they are moving in water, the eggs may or may not get fertilized. That is why fish and frogs lay hundreds of eggs. That is, it is not necessary or it is not possible that all the 100 eggs should be fertilized. It is only a chance of occurrence. I hope you understand. So this is, we talk about viviparous, oviparous, cridoid and non-cridoid eggs. Yesterday we have learned this. I didn't say about cridoid and non-cridoid. That you please understand. Are you clear? And then about the spawn I said. And then this, come back to sexual reproduction. Understand this. X, 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 Y. Just listen. This X, 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 Y. Okay. Then this is again X, 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 again X, Y. I hope you understand. It's solitary for function. X, 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 Y solitary. Right? We say the chromosomes, they occur in diploid. Is it not? We'll just continue. Just wait. We'll continue. So this chromosomes, we say they occur in diploid condition. So normally that diploid nature we represent as XX. Okay. And it is as diploid. This is also diploid. Okay. Now here this XX, it is representing female, XX representing female and XY representing male. XX representing female chromosome and XY representing male chromosome. That will present only in allosome. I hope you understand. We say allosome spelling patko. Allosome, it is a sex chromosome where XX female, XY male. I hope you understand. X chromosome, they are bit longer and straight in nature. As we know, chromosome we draw like this. Chromosome we draw like this. And male chromosome, it is one arm shorter, represented like this. So this is Y chromosome, X chromosome. Sorry, this is Y chromosome. This is X chromosome. I hope you understand. So now during the time of sex determination, takes place in the early development of embryo. First 3 months kulla sex of the child will be determined. This is XX female. Okay, and this is XY male. I hope you understand. It is in the female parent, male parent. As when this X is combining with this X, it will be XX. And say X combining with Y, it will be XY. So now this is female, XX female, XY male. XX female, XY male. So now this XX should be female, XX should be female, XY male. This is also female, this is male. I hope you understand. That is why we say Y chromosome is responsible for the, normally it is said, determination of the sex. The Y chromosome, vandana, it is male. Otherwise, in the X purnalo, it is as female. So that is how sex is determined. So I say XX female, female parent, and XY male parent. In that chromosome, male representing the sex is determined. I hope you understand. So this is called sex determination in man. Sex determination in man. Now what is the structure of chromosome? We know as centromere and then 2 arms. This is centromere and this arm is called pellicle. Centromere with 2 arms, that is called pellicle. And inside that we find a network called chromani matter. A network called chromani matter. And that in turn formed of DNA, in turn genes. DNA, deoxyribonucleic acid. And that DNA in turn containing genes. Genes are the carriers of hereditary characters. I hope you understand. So this is for the determination of the sex. So we say as structure of this formation of sperm and ovum by the process spermatogenesis and oogenesis. I hope you understand. So this is can you understand this? Then we will go to next. This is actually the introduction, right? So it's 23 parts, how it forms, the diploid, and how the sex is determined. That we are learning. Sure? And then this zygote as a result of fertilization. What is that? Fusion of male and female gamete forming zygote, and that zygote, it is diploid in nature. Okay? That zygote is diploid in nature. So now that zygote, this zygote will undergo further division, that is called cleavage. Zygote will undergo further division, that is called cleavage, right? And as a result of cleavage, the embryo called blastula is formed. As a result of cleavage, the embryo called blastula is formed. Okay, and that will go next cell division, that is called gastrulation, and the resulting embryo is called gastrula. I repeat, after fertilization, zygote is formed, diploid in nature, and that will undergo a series of cell division, which is again nothing but repeated mitotic cell division. And first it is cleavage, where it is forming 2, 4, 6, 8, 16, 32, 64, 128, like that. It multiplies till it forms irregular mass of cells called blastula. I hope you understand. Zygote, diploid in nature, is a single cell, single but diploid in nature, will undergo repeated mitotic division till it forms a irregular mass of cells called blastula. Okay, and that blastula, on further continuation of cell division, again will undergo mitotic division, and that forms the division called gastrulation, and as a result gastrula will be formed, and this gastrula is differentiating into 3 germ layers. Gastrula gets differentiated into 3 germ layers as ectoderm, mesoderm and endoderm. These 3 germ layers differentiating into outer ectoderm, middle mesoderm and inner endoderm. And this on further differentiation, it undergoes differentiation into various organ forming. So this organ formation, it occurs during the process gastrulation, cell differentiation. Yeah, so that is during gastrulation. And those ectoderm will give rise to certain parts of the body. Mesoderm will give rise to another parts of the body, and in endoderm will give rise to further parts of the body. Thereby differentiation of tissues will occur. And then next is organ formation. So this will be differentiation. This is as differentiation of organs, differentiation of organ, and then organ formation, organ system, organ formation and organ system, and then further growth will be developing. So till the month of 3, till 3 months, it is called embryo. The development of zygote till first 3 months, it is called embryo, and further development, it is called fetus. Further development, it is called fetus. I hope you understand. So this is further information or discussion about the lesson after fertilization. Okay, so the fertilized egg is diploid in nature, but it is a single cell. Are you clear? So next one we say as zygote formation. Yeah, we know about gametogenesis, spermatogenesis and children's. Right? Now during this viviparation, where we come across a term microlecythal, megalecital microvesicle, megavesicle, and avesicle. Microvesicle, megavesicle, and avesicle. What are they? They are the fertilized eggs. They are the fertilized eggs. Okay. Now, what is the content of the egg? Egg nothing but the developing embryo and the nutritive tissue. Developing embryo and the nutritive tissue. Nutritive content we say. Now, this nutritive content, nutrient, they are nothing but yolk. The nutrient contents, they are nothing but yolk. Now, when this yolk, other than this embryonic portion, when it is very meagerly, very poorly developed, distributed, it is said to be microvesicle. When the yolk content or nutrients, when they are poorly developed, they are called microvesicle. When they are distributed in abundance, it is said to be megavesicle. All the birds, they are megavesicle egg because they contain rich nutrients. Especially in birds, hens egg padikkuromla, as nutritive content, yellow yolk and white yolk padikkuromla, they are abundant in nature. So it is megavesicle egg. And when there is no yolk at all, it is said to be avesicle. Okay. So this micro, it is very meager, less amount. It is large amount, large yolk. It is no yolk at all. Avesicle, no yolk. Then how do they allow the embryo to develop? They are by the presence of placenta. They are by the presence of placenta. Placenta is the embryonic connection, vascular connection between the developing embryo to the mother. That is placenta. I hope you follow this. And then we learn about egg membrane. We learn about egg membrane. We are about egg membrane as egg membrane. We learn terms. One is zona pellucida, corona radiata, zona pellucida, corona radiata, and vitelline membrane. Zona pellucida, corona radiata, and vitelline membrane. This is corona radiata. Okay, and then inside we find okay. So this is pellucida, zona pellucida. Outer corona radiata. Inner to that is the nucleus and cytoplasm. And in the third layer, it is the vitelline membrane. I hope you understand. So this is structure of a vesicle egg, where it has outer corona radiata, middle zona pellucida, and innermost vitelline membrane. I hope you understand this. So this is for the structure of human ovum and egg or egg, and it is a age-cycle time. I hope you understand. So this is for our second part of chapter 9, Reproduction in Animals, as video 7. We will continue in the next video session. Have a good day. Thank you.